My pleasure to introduce our next speaker, Paul Turner. We'll be speaking about Paci or Paci?
Paci-PHI.
Paci-PHI. Yeah, sorry. I knew that. Thanks, Paul.
Sorry, thank you. Good afternoon. I'm here to present Paci-PHI, which is the next generation in antimicrobials. We know that the global disease burden of antibiotic-resistant bacteria is huge in both the developing and the developed world. This is arguably the largest public health crisis that we have in the current century. Our solution to this problem, and it's a very familiar problem, we'll start with the problem. So by the time a new antibiotic goes to market and all of the millions of dollars spent on it, the expectation is that you can have failure. So if you look around the world in terms of resistant isolates of bacteria that are able to withstand this drug, this is essentially what we found over and over and over again. A projection map like this one on the right, which was alluded to in an earlier talk, is that by 2050, we'll have millions of deaths around the world, both the developing and the developed world, especially in the centers of the world with the highest human populations. So another solution should be found. There is an old idea that could be updated, and that is to use bacteriophages, which are viruses that are specific to bacteria. So in the upper left, I have a very familiar replication cycle of a lytic phage, where here you can consider this to be a self-amplifying drug. The ability of this phage to target specific bacteria, amplify within the cell, kill the cell, and then move on to destroy cells of similar type. Phage therapy is safe. Phages are safe for ingestion. I will talk about that more in a moment. But there's an obvious problem. Just with antibiotics, bacteria have many, many ways of evolving phage resistance. So our innovation is to find phages that exert the right selection pressure on bacteria such that if they bind, enter, and kill, they're doing their job. And as a prediction, as an evolutionary biologist, I'll tell you that the bacteria will respond. And the idea is to find a solution that is evolution-proof, such that when the bacteria respond through evolution, they become more vulnerable to something that is already pre-approved, such as existing antibiotics. In this diagram in the middle, it illustrates the point for the candidate specific bacterium for which we've made the most advancement, and that's Pseudomonas aeruginosa. It's famous for having efflux pumps all around the cell, which take antibiotics and move them out of the cell. And this works across a broad array of antibiotic classes. We have found phages here in Connecticut that bind specifically to the outermost proteins in these efflux pumps. These proteins are highly conserved. Bioinformatics analysis shows us that, so that it is not surprising this works on a very, very wide variety of genotypes of this bacterium. So essentially what will happen is it paints the bacteria into a corner. They will die from phage attack. If they evolve resistance to the phage, they are unable to move those antibiotics out of the cell. So we have made a lot of advancements in this system with our first target of Pseudomonas aeruginosa. Plenty of in vitro data showing that when the phage exerts selection pressure on the bacteria, they become sensitive to existing antibiotics. Mouse model data show us for acute pneumonia, the phage plus antibiotic is able to restore and rescue the life of the mouse when the antibiotic alone won't do the job. Most importantly, we've tried this in 2 emergency cases with human patients. In January 16, an elderly man, we were able to treat his biofilm infection on an artificial substrate of his heart and aortic graft. And more recently, in December 2017, we added phages to the nebulizers of a CF patient in Texas, and we were able to resolve her lung infection. And just like we saw in the laboratory, these very many drugs for which she was pan-drug resistant to, after the therapy, we not only reduced the bacterial load so that she was healthy, but all the remaining bacteria were changed to sensitivity. So now her physicians have plenty of options for treating any problems. Last, I will say our intellectual property has been helped with Yale to be secured. We have ambitions for trying this in the CF-associated pulmonary infection clinical trial. And we have very, very many other phages that do a similar analogous, causing the bacteria to become less virulent or to become sensitive to antibiotics, and a large number of other target cases. Our funding model is the following. We have discovery and characterization, optimization and testing, manufacture and distribution that would, to the tune of $3 billion, get us to where we want to be. But immediately we're seeking funders and partners for our immediate goal of trying this in a clinical trial. I'll end with our team that has nearly 3/4 of a century of combined expertise in basic bacteriology and biomedical microbiology, industry experience, as well as biomedical and surgical experience. So we feel like we're the right team to do the job. Thank you for listening.
Great, Paul. It's so creative and so exciting. Questions from the audience? In the back.
Do you know if the human immune system will attack the bacteriophage if you're using some delivery system other than a nebulizer, like we were just talking about?
So you're asking whether there's an immune response at all to phages?
Yes.
Yes. So there is a small, sometimes detectable immune response, but you see phages every day. Probably during the course of my talk, you've ingested them or taken them in through dust particles that they're adhered to in the room. So humans don't mount a big response to them, and we have almost a century of data to tell us that. We're looking at exactly how the immune system works with this system, but we don't anticipate any problems.
Question back here.
Dr. Turner, does each phage have to be personalized for each infection?
No, importantly, what we found in these two cases and what we've seen in the laboratory is that as long as we are targeting a highly conserved protein in terms of binding, there's no problem in terms of exact specificity.
One last question. One up there.
If you're targeting a conserved efflux pump, how are you able to separate pathogenic and commensal bacteria?
The easiest way to do it is go after something that has no business being in the human body. And if you have specificity to the outermost protein in those efflux pumps, that would be perfect. Similar would be in TB. So I agree it might be more difficult in the other systems when we are developing protein specificity, but I didn't have time to go into the details of those aren't efflux pump protein specificities. That's more like virulence proteins associated with pathogenic E. coli that differ from the proteins of your normal flora.
So you still can't use it for Neisseria?
No, it's jumping between species is difficult, but I would say that our pipeline to discovery and characterization gives us optimism that we could design it to almost anything when drawing upon the vast biodiversity of the phage world.
Great. Thanks so much, Paul.
Thanks.